let us get started. Uh, Cell is the main building block of human beings. Like human beings are composed of millions and trillions of cells, and cells compose tissue, and tissue form organs, and organs form organ system, and an organ, different organ systems are what constitute a complete human being. So basically, a cell is what has made us a human, starting from a cell and then a tissue and then an organelle and then organs and then organ system and then a human being. This is a hierarchy which has put us to our fully developed form as human beings. So my first slide says, what is cell? Cell is a basic unit of life. All chemical and physiological and physical reactions that make us alive happens in the cell. For example, the cells in our stomach, they secrete acid to digest the food. The cells in our mouth, they secrete saliva to secrete salivary secretions. The other cells in our skin, they protect us from the invading pathogens. And the cells, for example, in our nails and our hair, everything is composed of cells. So all cells arise from existing cells. It's the development of cells. Like one cell divides into two, and then two cells will divide into four, and they keep going dividing. And a cell is a building block for all living organisms. And cell organisms are unicellular or either multicellular. Unicellular means they are composed of only one cell, and multicellular means like us, like human beings. Okay. Okay. So multicellular means they are composed of more than one cell. And each type of cell has its own specific function, like I had already elaborated. Just like cells in stomach, they secrete enzymes which digest our food. And cells in our brain, they are neurons. They transfer information from one neuronal cell to the other neuronal cell. Similarly, cells in our kidney, they reabsorb the fluids and water which makes the kidney function. Cells in our liver, they modify and they digest and they do chemical reactions with fat. So every cell has its different role. And just like immune cells, they fight against pathogens. Pathogens are like microorganisms like bacteria or viruses. So I just wanna elaborate that cells have their different functions. Each cell is supposed to do a different function. Uh, Next slide. I would like to. Okay. So how do we study cells? We have to use a microscope to study cells because we cannot see cells with naked eye. So we use different kinds of microscopes to study cells. The minimum magnification microscope is a dissecting microscope, which magnifies a cell to 100 times of its original size. And then comes a light microscope, which magnifies a cell to 1,000 times of its original size. And the most magnification is provided by electron microscope, which is used in labs of higher level of labs. There are two microscopes shown here. So this one is the light microscope and the other is the dissecting microscope. These are the microscopes which are commonly used in our labs, in our schools, in our colleges, and in our universities, and every common place where we can go. So these microscopes work on the principle of magnification. There are some lens physiology which we do not need to discuss here. Most light microscopes used in college biology lab can magnify cells up to approximately 400 times. Because you see, these are mostly light microscopes. Dissecting microscopes have a lower magnification, which we already read in the previous slide. Like they have 100 time magnification power. And dissecting microscopes are used to examine larger objects such as tissues. Because you see, higher the magnification power, the smaller size we can see, and lower the magnification power, the larger the object needs to be in order for this proper visualization. I just wanted reassurance. Can you guys still hear and understand what I'm saying?
Yes, Dr. Muhammad, very clear. Very interesting
All right,
pictures. We can see very nice pictures there.
all right, all right. So there are
Fantastic.
two pictures which compare light microscope versus electron microscope. So a light microscope, as we already know, it magnifies up to 100 times of a cell's original size. And an electron microscope, which is a big, big beast, it magnifies up to like 100 million times of a size, original size. So on the left, we can see a pink histological image in which the darker pink areas, like these, if you can see my pointer, these are the bacteria. These. This is a bacterium and this is a bacterium. This is taken with light microscope. And on the right, there is a more magnified image taken from electron microscope. This shows the bacteria. Now you can differentiate between the magnification and all the details of the bacteria from electron microscope, but you cannot do so with light. So the example shown here is a Salmonella bacteria. Have you ever heard of typhoid fever? In typhoid fever, the responsible bacteria which causes typhoid is Salmonella bacteria. So this is a picture of Salmonella bacteria taken with light microscope and taken with electron microscope. Here it is shown some approximate cell sizes and the comparison of an atom, then a lipid, then protein, as compared to other things. For example, the smallest thing to ever exist in this planet is an atom. And then there are lipids. Lipids are molecules which are composed of different kinds of atoms. Then proteins. Proteins are larger than lipids, and bacteria are even larger. Then comes the mitochondria. We will explain in detail later what a mitochondria is. And this is just a comparison of different sizes. Like you can see from 0.1 nanometer, then 1 nanometer, 10 nanometer, and so on for 1 meter for an adult female. So
Well, we have now transistors, I think, by Intel AMD computers, where the transistors wires, I believe they've gotten them down to 4 nanometers. So
yeah,
very, very
that's cool.
interesting that we're getting to the kind of the cellular level. We're just right between the
Yeah,
around a lipid and a protein.
yeah, cool. Then I would like to teach you cell components and types. So cell is also composed of certain components. Components which form cells. A cell is like a single factory. You see a factory where different machines are there and different products are being made by different machines. So a cell in itself is not producing anything. There are different components, there are compartments within the cell which give rise to different products. For example, if there is a cell and for example, we have a thyroid gland in our neck. You see, there is a thyroid gland in the middle of our neck which secretes certain hormones called thyroid hormone. So there are specific kinds of cells within the thyroid gland which secrete thyroid hormone. And there are also other hormones like calcitonin which are also being secreted by the same gland. So it's the same gland, but there are different types of cells and different compartments within the same cells which produce different hormones. I hope you get my point. So cell components are cell membrane, which covers the cell, and then the cytoplasm, which contains all the organelles and all the material of a cell, and then comes the nucleus, which contains the DNA genetic information of a cell. So there are cell types, different cell types. One is the prokaryotic cells, and the other is the eukaryotic cell. A prokaryotic cell is without a nucleus. However, it also have DNA, but it does not have a nucleus. The distinguishing feature between a prokaryotic cell and a eukaryotic cell is nucleus. Whenever you get a cell and there is no nucleus in it, go for prokaryote. And whenever you get a cell, there is a proper well-formed nucleus with a covering membrane. It must be a eukaryotic cell. So eukaryotic cells are what we form all of us, like animal cells and plants are the eukaryotic, but prokaryotic cells like bacteria. So there is an image shown of prokaryotic cells and different parts of a prokaryotic cell. So there is a cell membrane, there is flagellum, there is cilia, there is no nucleus, there is just a genetic material dispersed like a thread in the center of the image you can see here. This is the genetic material. But in contrast, this is the eukaryotic cell. You can see there is a fully formed nucleus within the center of the cell. So that distinguishes a prokaryotic cell from a eukaryotic cell. And there are other well-formed organelles which are going to study one by one. So now coming to the eukaryotic cell, it has a plasma membrane, it has a nucleus, it has a cytoplasm. The eukaryotic cell is a proper cell. It has everything in it required for the proper functioning of a human body, for the proper functioning of an animal body, for the proper functioning of plants. So eukaryotic cells has to be carrying all those organelles, all those cells, and all the nuclear material in themselves. So it is composed of plasma membrane, and in case of cell wall, in case of plants, there is no cell membrane, there is a cell wall. Plants are having cell wall plus cell membrane, but animals do not have a cell wall. And the reason why animals do not have a cell wall because animal cells needs to move. Animal cells need motility, but cell wall is a rigid structure. So if animals would have cell wall around their cells would not be able to freely move. That's why we humans and all other animals they lack cell wall around their cells, but plants do have a cell wall. And then comes the nucleus, which is the genetic material containing organelles. Then comes the cytoplasm. Cytoplasm is the fluid-like material within a cell that contains all the organelles. So I have named some of the organelles here. First is mitochondria, then ribosomes, then endoplasmic reticulum, then Golgi apparatus, then lysosomes, then peroxisomes, then cytoskeleton, then vacuoles, and then chloroplasts. We will be going in detail of these one by one. So, doctor, do you have any questions?
Hi, yes, Dr. Mohammed, thank you so much. Yeah, I just had a quick question regarding the cell wall of plants versus humans not having a cell wall. I know in human tissue, typically, for example, in skin cells, the cells start in the deeper tissue and they work their way kind of towards the surface. I'm wondering if that's the case in other organs or parts of the body where the cells kind of move from the center or core outwards. And if that's the reason why plant cells can have the rigid cell wall, or do plant cells also grow that way, meaning that they start from the center and they push their way out? Is there a different
Okay, I got
mechanism?
your point.
Yeah, because in other
There
words,
are,
the skin cells of a human theoretically could benefit from having, the outer cell having a like a cell wall, but
okay,
it would prevent the motion of the cell from deep to out of the tissue.
fine. So nature has devised a mechanism for human skin. They do not have a cell wall, but they do have keratinization. We do have thick keratin layers over our skin. You see the outer surface of the skin. This is not skin cells. This is the keratin on our skin. The same keratin which is deposited abundantly in our nails and our hair. So keratin is what provides protection to the skin cells, and that's why they do not need a cell wall. So the mechanism which you are asking regarding the movement of cells from like deeper to the outer layer, that is true for skin. But see, for other organs like stomach, like liver, like kidney, cells always proliferate and divide and then move where they are needed. For
Ah,
example, in our blood, there is an injury somewhere on your body. So we need platelets over there. We need fibroblasts over there. We need some infection fighting cells over there. So all the cells will keep on moving toward that particular area where the injury is happening. So you see, it's always need based for all other organs, and for the skin, it is obviously the one going from deeper to outer layer.
so a plant cell, when it appears in a certain part of the plant, it cannot move. It stays fixed wherever it emerged or wherever it's split. It just
Yes,
stays there. It cannot move
it
anywhere.
just
Got
stays.
it.
It cannot move anywhere. Yeah,
Interesting.
sure.
Okay, fantastic.
Yeah, thank you.
Students, if you have any questions, please raise your hand, and at an opportune time, Dr. Mohammed can call upon you if he sees your hand.
Okay, so I guess we should keep on moving.
Yes, please.
Okay, so the first organelle we're going to study is the cytoskeleton. As you see the word skeleton tells us that cells do also have certain kind of skeleton which holds them together. I already told you that you imagine a cell as a factory. So in factory you need building blocks. In factory you need pillars. You need a building which keeps the factory intact. So cells have that pillars, have those building blocks, have those bricks in the form of cytoskeleton. A cytoskeleton is composed of three things. Those are microtubules, intermediate filaments, and microfilaments. You do not have to go into the deeper details of these tubules. Just remember the name. The cytoskeleton is composed of microtubules and intermediate filaments and microfilaments. And from the examination point of view, they may ask you what are microtubules formed of? They are made of tubulin protein. And what are microfilaments are made of? They are made of actin protein. So tubulin and actin are different kind of proteins which make up the cytoskeleton of a cell. So what does cytoskeleton function with? It helps with the movement of the cells. It helps in the contraction in the muscle cells and cleavage growth during cell division. So when a cell divides, it just divides. One cell is divided into two. A cell is divided into half, and it is pinched in the center. So whenever there is a pinching, as we were already discussing about the skin cells, they divide and they move toward other areas. So whenever there is like cleavage of the cells, cytoskeleton assists that cleavage of one cell dividing into two cells. So these are certain functions of cytoskeleton. Basically, it helps with the movement and just like when we contract our arm, so our bicep muscles contract. That contraction is done by cytoskeleton. These are some images showing microfilaments, intermediate filaments, and microtubules. This is like a skeleton. You see a skeleton of a human. We have all the bones and ribs and cervical spine and a vertebral column. In the same way, this is a skeleton of a cell which is holding this cell together. Otherwise, the cell would just fall apart. And the below image, we can see a cell dividing. So this was a whole genetic material of a cell. It just got divided into two. And this division was brought about by microfilaments, microtubules, and intermediates. So that was the function of cytoskeleton, to divide a cell and to maintain the integrity of it. Next come the two important organelles, those are cilia and flagellum. Not all the cells have cilia and flagellum. These are the specific kind of, you can see these are the limbs of a cell. These are like arms and legs of a cell. So the cells which require more movement, they require cilia and flagellum. And the cells which do not require any kind of particular movement, they do not require cilia and flagellum. These are particular hallmark of bacterial cells. So bacteria do have cilia and bacteria do have flagellum because they need to move from one place to another and spread the infection. So cilia are they are small in size and they are many in number. They are short, they are hair-like, and they help in movement of cells or substances. Flagellum are like lesser in number as compared to cilia, and they are very long, and they also help in the movement. So basically, the function of cilia and flagellum is the movement of a cell. The difference is in their structure. Cilia are short and hair-like, and flagellum are long and they are thread. Then comes the nucleus. Nucleus is the central core of a cell, just like a brain of the human body. A nucleus is the brain of a cell. So its structure is a nucleus has a nuclear membrane. If I am able to go to the previous slide here, we can see a nucleus here. This is the nuclear envelope outside the nucleus, the light pink color. And then in the center, there is a nucleolus. And then there is the genetic material residing inside the nucleus. So you can see the structure of a nucleus. The nucleus consists of nuclear membrane and the nucleoplast. The nuclear membrane is the envelope of the cell which has nuclear pores. What does nuclear pores do? They allow passage of some substances from the nucleus in and out. So nucleoplast, which is inside the nucleus, it contains the DNA. So in this image, as you can see again, this is the nuclear envelope, and these are the pores visible in it. And this light purple is the nucleoplasm, which has a DNA, and this dark brown color is a nucleolus. This is also a diagram of nucleus showing you a nucleolus and chromatin material and nucleoplast. Then comes a group of organelles which are collectively called as endomembrane system. So endomembrane system is a system of structures of different shapes made of membranes. Its role is in making and modifying proteins and lipids. It includes plasma membrane, nuclear envelope, rough endoplasmic reticulum RER, smooth endoplasmic reticulum SER, Golgi apparatus, and lysosomes. So these are the organelles. These are different organelles performing different functions, having different structures, and they collectively are called endomembrane system. Now, why do they are called endomembrane system? system, because endo, they are inside the cell, and membrane, they are the membrane. So I would like to have some questions.
Yes, we have a question from Thabisli Mgom, Mgomez. Thabisi asks, he wanted to find out the difference between lysosomes and peroxisomes.
Okay, peroxisomes are the organelles, and lysosomes are also the organelles, but the difference is in their function. Peroxisomes are supposed to detoxify the cell. Peroxisome, there are certain molecules like hydrogen peroxide, which are produced inside the cell, and they are toxic to the cell, and they need to be eliminated from the cell. So what does peroxisomes do? Peroxisomes clear that hydrogen peroxide like toxic substances. What lysosome does, they phagocyte those different substances. They divide and they clear the cell's debris and cell's necrotic material. You see, cells are always evolving. There are certain organelles are formed and certain organelles are destroyed. So the ones that are destroyed needs to be eliminated from the cell. So lysosomes does that part.
And Dr. Mohammed, there's a lot of talk about the process of fasting, and we're in Ramadan right now, so a lot of
Sure.
people around the world are fasting. There's certain processes that are activated in the body to kind of kill zombie cells, to cleanse the body. Does the process of fasting increase the activity of peroxisomes, or what type of
Lysosome.
things? Oh, lysosomes. Okay.
Yeah, it does increase the activity of lysosomes because lysosomes help to digest the dead and debris part of the cell. So yeah, lysosomes activity is increased during fasting. And I
Fantastic.
guess we do not have to go to the in the detail of that process. Just we have to know that lysosomes are what get activated. So coming back to our point, we were studying endomembrane system, and I would like to go back to this diagram. The very pretty picture. I would like to explain all the organelles from this diagram. So there is, you can see an endomembrane system organelle. What is this? This is endoplasmic reticulum. And there are certain black dots over here, which you can see. This is the rough endoplasmic reticulum. And on the inside, this is the smooth endoplasmic reticulum. I'm just making you guys well acquainted with the organelles and their location inside the cell. So this is a endoplasmic reticulum. Outer side is which have a black bead-like structure. This is rough, and the inside is smooth. Then comes this part. This is the Golgi apparatus, and this is the mitochondria. This is not included in endomembrane system. This whole structure, the dark blue, is the plasma membrane of the cell. And these here you can see also cytoskeleton. So we will be focusing on endomembrane system organelles. These are one endoplasmic reticulum, then Golgi apparatus, and then a plasma membrane. And I guess these are the lysosomes or peroxisomes or whatever they are. This diagram. The endomembrane system works to modify, package, and transfer lipids and proteins. Endomembrane system is particularly related to the package and transport and modification of lipids and proteins. So you can see Golgi apparatus and endoplasmic reticulum are those machinery which are installed in a factory to make different products. Endoplasmic reticulum have ribosomes on it. Ribosomes make proteins, and those proteins are then secreted inside these canal-like structures of an endoplasmic reticulum. You can see like in the middle here, and then from here they are pinched out of the endoplasmic reticulum in the form of flaps, in the form of a sac, in the form of some containing, you can say, containing box, and then they are transported to Golgi apparatus. Then Golgi apparatus takes these proteins or lipids and it modifies them. It, you can say, packs them. It just does some finishing makeover touch, and then it exports them out of the cell. So this is the mechanism by which endomembrane system works. Endoplasmic reticulum is made up of membranes as boundary. It has membrane around it. There are two types, which are rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum has ribosomes attached to their surfaces on the membrane. It is made of flattened vesicles and attached to nuclear envelope. Its role is in modifying protein. So as you can see in the previous picture, this was the nucleus, and the endoplasmic reticulum is attached to the nucleus, and these little bead-like structures are the ribosomes, which secrete protein. And then this endoplasmic reticulum helps in secretion of proteins, and these proteins are secreted in the Golgi apparatus. So then we have a smooth endoplasmic reticulum. Smooth endoplasmic reticulum is the part of endoplasmic reticulum which does not have those ribosomes, which does not have those various structures attached to it, like you can see here. This is a smooth endoplasmic reticulum, and the function of smooth endoplasmic reticulum is also some kind of modification of protein molecules and some detoxification of toxic substances. Its role is in storage of calcium ions. That's a major function of the endoplasmic reticulum. It stores calcium ions. It synthesizes lipids and steroids and carbohydrates. So this is all about endoplasmic reticulum. Then comes the Golgi apparatus. Golgi apparatus, as we have seen here, this is the Golgi apparatus. Golgi apparatus takes certain lipids and proteins from endoplasmic reticulum. It modifies them and it then excretes them out of the cell. Golgi apparatus has flattened big vesicles or sacs. So the structural difference between endoplasmic reticulum and Golgi apparatus is the size of its sacs. You can see its sacs are small and Golgi apparatus has larger sacs. It has two phases. One is the trans phase and one is the cis phase. So you can see this is the cis phase, which is towards the nucleus, and this is the trans phase, which is towards the cell membrane, which is more close to the outer surface. Function is modifies proteins and lipids that I have already told you. This is some electron microscopic image of a Golgi apparatus. You can see here, these are the sac-like structure which you can appreciate here. This is the Golgi apparatus. And what again will be the cis phase? The cis phase will be the one facing nucleus, and the trans phase will be the one facing facing plasma membrane. Then we have lysosomes in endomembrane system. Lysosomes are the structures which are membranous vesicles. So these are not like long sacs. These are simple rounded vesicle structures. They contain enzymes and they have low pH. Low pH means they are acidic. So as I already told you when a doctor asked me about the effect of fasting on lysosomes. So lysosomes clear out the debris from the cell. They clear out the dead and the destroyed parts of the cell. So for that, we need acidic. So there is a specific pH for specific functions, and for the clearing out of some debris from the cells, there we need low pH. So lysosomes have low pH and they contain different enzymes to dissolve the dead parts of the cell, and so that they can be excreted out of the cell. Their role is as garbage disposal. As the name can clearly show you the function of lysosome, they dispose garbage by removing any broken organelles or pathogens, etc. In macrophages, a type of white blood cells. So macrophages are a kind of cells which are a type of white blood cells that help provide us immunity. Whenever there is any bacteria or virus inside our body, macrophages take that organism and they kill them. How does macrophages kill bacteria or viruses? Macrophages have a lot and lot of lysosomes. And what does lysosomes do? They secrete enzymes and they have acidic pH, which helps in the killing of bacteria and viruses taken up by macrophages. So here you can see a lysosome. This is a lysosome, and this is some dead part of the cell which needs to be cleared. So this dead part of the cell will come near the lysosome, and there will be fusion between lysosomal membrane, this darker pink membrane, and this darker blue membrane. This membrane is of the cell which need to be cleared, and the lysosome and the cell will then fuse and become a single. You can say the two cells combined and form a single cell. It's not like we can just reformulating a big cell. It's like we are just fusing two different cells so that lysosome can release its toxic enzymes inside the cell, and this can be cleared. So here we have a lysosome which contains a phagocytosed material, which is now will be cleared by the enzymes of life. A macrophage has phagocytosed a potentially pathogenic bacterium. So that particular cell we were talking about was a macrophage, which has a this darker brown colored bacteria into it. So a macrophage took up some bacteria and it then fuses with the lysosome within the cell so that the pathogen can be destroyed. Other organelles are present in the cell, but for simplicity are not shown. So if one may ask that why is this cell so simple? So the answer is just because it is not shown due to the simplicity. There are also other organelles, nucleus, there is Golgi apparatus, there is endoplasmic reticulum also flowing around in the cell. So a cell is like a river, and in a river we have dolphins, we have other fish, we have small fish, we have like scorpions, we have like crabs, but everything is like floating in that ocean. So this is how a cell works. It has different organelles which keep on floating inside it. But for the sake of simplicity, we have shown only two things. One is the macrophage and the other is the lysosome. Then come the ribosome. We have studied earlier ribosome that ribosomes are what? They are present on rough endoplasmic reticulum. And on rough endoplasmic reticulum, what does ribosomes do? They produce proteins. Ribosomes are the organelles of the cell which have no cell membrane around them. Ribosomes do not have any kind of membrane around them. They are simple structures which has larger subunit and a smaller subunit, and these two subunits combined form a ribosome. So these are the organelles which are not made of membranes. They are made of ribosomal RNA and proteins. What does ribosomal RNA mean? Ribonucleic acid. There is a DNA which means deoxyribonucleic. Attached to the endoplasmic reticulum, as we already discussed, that ribosomes they also present free in the cell, but most of the ribosomes are attached to the endoplasmic reticulum. They can be free in the cytoplasm. They are round in shape. They function in synthesizing protein. This is all about ribosomes, and then comes the mitochondria. So mitochondria is the powerhouse of the cell. So we call them powerhouse or energy factories. Cell does a lot of functions during its lifetime. A cell form organelles, a cell form proteins, a cell form lipids, a cell shares genetic material in the form of DNA. For all those purposes, a cell needs energy. We all need energy, and our cells also need energy. And where does that energy come from? It comes from mitochondria. Mitochondria produce energy. It functions in making ATP. Adenosine triphosphate is a chemical compound. It is a form of energy, and it comes from food. Structure of mitochondria. It is oval shaped like this. It is oval in shape. It is not rounded. It is made of two membranes, an outer membrane and an inner membrane. So I would like to ask a quick question here. What other organelles did we study previously which had two membranes, like an outer membrane and an inner membrane?
Okay, so we have a pop quiz.
Yep.
Anybody want to take a try at an organelle we reviewed with two membranes, a double membrane?
Okay, so I'm gonna answer it myself. That was nucleus. It has a nuclear envelope in the form of two nuclear membranes. One was outer membrane and one was inner membrane. And now we have the mitochondria, which also has an outer membrane and has an inner membrane. The inner membrane has folds which hold cristal. Like this folding membrane is the inner membrane of mitochondria, and these folds, these depressions, these pits, they are called cristal. And then there is intermembrane space like this. There is a space between the two membranes. They are not fused together. And then we have the matrix. Matrix is just, and then matrix is what? Matrix is the fluid which is present inside the mitochondria. And there was a kind of matrix inside the nucleus which we call nucleoplast. And then there was a kind of matrix inside the whole cell in which the organelles are present. We call that as cytoplasm. So different kind of matrix are like different kind of fluids which in which different substances are dissolved for the proper functioning of that organelle or cell.
Professor Mohammed, one question regarding the mitochondria.
Sure.
Oh, maybe you're going to explain it. I was going to ask a little bit about the electron transport chain. Um, you know, apparently the mitochondria takes electrons that are higher valence, like higher energy levels, and somehow is able to lower their valence to take the energy out of those and use it to form, sugars or ATP. Um, how do we know how that actually happens in the cell, and what's the similarities or differences versus like a electric current, this electron transport chain?
So we have an electron transport chain working inside the cell and particularly working inside the mitochondria, which helps us produce ATP energy. And that is a complete detailed topic which we may discuss sometimes later. But I will
Okay.
just, in the next lecture, we will be discussing electron transport chain. I showed you that. But for the now, that for the time being, you should just know that electron transport chain uses NADP and it uses other molecules which receive that electron and then they pass on from one molecule to the other molecule. There are different molecules which are called cytochrome, cytochrome A, cytochrome B, cytochrome C. And the electrons are taken from one molecule, let's say cytochrome A, and then they are passed to cytochrome B, then they pass to cytochrome C. And finally, when they are given to hydrogen and oxygen, they form water, and as a byproduct, energy is produced. So this was an overview of electron transport chain. Now, there we have a transmission electron micrograph which shows a mitochondrion as viewed with an electron microscope. Notice the inner and outer membranes, like this is the outer membrane, and this inside is the inner membrane, which is like distorted membrane. The cristae are these depressions inside the membrane, this, all right, this, and the mitochondrial matrix. The whole, everything inside is the mitochondrial matrix. Now comes the organelles, which are not a part of the human cells. They're a part of the plant cells, and these are chloroplasts. Chloroplasts are vesicles with two membranes. So we had nucleus with two membranes, we had mitochondria in two membranes, and now we have chloroplasts with two membranes. And chloroplasts are present in plant cells, not animal cells. They have inner and outer membranes, and they have stroma. Like their matrix is called stroma. It is inside the chloroplast. They have thylakoids, which are flattened vesicles inside the stroma. The thylakoids are organized in stack form, which are called grana. I will be elaborating this with a diagram later on. And these chloroplasts organized, these grana are made of membranous boundary, which contains green pigment. You see all that greenery out there, all the plants are like green. So that green color is due to the chloroplast because chloroplasts have green pigments inside them, which are called chlorophyll. And the lumen of the chloroplast, what does the chloroplast do? Its role is in photosynthesis. You already know about photosynthesis. It is the process by which plants produce energy. It transforms solar energy into food, which is chemical energy. So this is the diagram of a chloroplast. This is the outer membrane. This is the inner membrane. These are the grana. These are the granum, singular granum and plural granum. These are stacks of thylakoids. These are like you can see there are different bricks on top of each other. One single brick is called thylakoid, and when different bricks are stacked over one another, they form grana. So this is the functional unit of a chloroplast. And why it is shown green? Because they have green pigment, which is chlorophyll, and which is necessary for the production of food. So chloroplast helps plants in photosynthesis. Next comes the extracellular environment. So we discussed various organelles of the cells, like nucleus, like mitochondria, like lysosomes, like Golgi apparatus and endoplasmic reticulum, ribosomes. Some were producing proteins, some were producing lipids, some were modifying and transporting those proteins and lipids. So where does those proteins and lipids go? Where do they go when they leave the cell? They go into the extracellular environment. It is the environment in which a cell resides. So, what does an extracellular environment do? It gives you protection against tension and compression, which is provided by cell membrane, or in case of plants, it is provided by cell wall. Extracellular matrix is what composes extracellular environment. It is the fluid which is present around the cells. This is called extracellular matrix. In animals, it is made of protein, collagen, and glycoproteins. Glycoproteins are macroproteins like bigger size proteins which are composed of sugars and proteins. And we have collagen. Collagen is like a thread-like structure which gives structural integrity to extracellular matrix. Just like we had our intermediate filaments or we have our microtubules, same as collagen provides skeleton to the extracellular environment. In case of plant cells, we have cell wall, which is made up of carbohydrates like pectin. So here is shown some extracellular matrix. See this. This is the boundary of a cell, which is cell membrane. Cell membrane is shown over here, which has lipid bilayer. We are going to discuss cell membrane in the later slide. So this is the cell membrane. This is inside of the cell, and these thread-like structures are what? They are intermediate filaments. They are tubulins. They are microfilaments. They are cytoskeleton of the cell. So this is the cell's internal environment. This is the cell membrane, and this is the extracellular environment. An extracellular environment is composed of collagen fibers like these. These are collagen fibers. It also contains fibronectin, and fibronectin is composed of the extracellular matrix, which consists of network of substances secreted by cells, just like polysaccharides. So different substances are formed in the cell, like they are formed in the endoplasmic reticulum. They are being picked by Golgi apparatus, then they are transported via cell membrane outside of the cell, and then they give rise to different structures, including fibronectin. And fibronectin, along with collagen fibers, they form the extracellular matrix. Cell connection. So different cells are connected to one another via cell connections because they do need to be in touch with each other in order to function properly. Just in a few minutes ago, I gave you an example of thyroid gland. So a thyroid gland is not a single cell; it is composed of various number of cells and millions of cells which function as a single gland. So when one cell secretes a hormone, it is passed to the next cell. So for that transport to happen, cells need to be connected. So connections are what holds the cells together. They can be tight junctions, they can be desmosomes, and desmosomes and tight junctions are different kind of cell junctions communicate between cells through exchanging materials. So, as I already discussed, we need some kind of channel between two cells in order for the exchange to happen. So, there can be plasmodesmata in plants and gap junctions in animals. So, we have just studied different kinds of cell connections. They can be tight junctions, there can be desmosomes, there can be plasmodesmata, and there can be gap junctions. These are just various kinds of cell connections. Okay. So there are four kind of connections between cells. So we have the little bit of detail of different cell junctions we studied here. There we have a plasmodesma. A plasmodesma is a channel between the cell walls of two adjacent plant cells. So this is an image of two plant cells connected to each other. And there you can see is a channel which is called plasmodesmata, which allows the passage of water and other molecules from one cell to the other. And then next we have tight junctions. Tight junctions are usually in for the cells which need a proper system of transport from one cell to the other. So here you can see this is a cell and this is a cell, and then there is a tight junction between two cells. And then we have desmosomes. Desmosomes are like some thread-like organelles and some pores which through which some different thread-like organelles are passed. So we have desmosomes, like desmosomes of a cell here, and there are intermediate filaments of cell there. So this is a cell, this is cell's internal environment, and then this is the membrane of a cell, and this is the membrane of the second cell, and this is the internal environment of the other cell. So this is the extracellular matrix here. This is one cell, this is the other cell, this here, right here, in between the two membranes is the extracellular material where desmosomes are present. And desmosomes are what? They are channels, they are pores, they are like little pores in the membrane of the cell which connect one cell to the other. And then come the gap junction. Gap junction channels are like this. There is this membrane of one cell, plasma membrane of cell number one here, and then plasma membrane of cell number two here. And connecting these two membranes is the gap junction. So these are four kinds of connections between the cells. One was the plasmodesmata, which is in the plant cell. Other was gap junction. Then comes the desmosome, and then comes the gap junction. A plasmodesma is a channel between the cell walls of two adjacent plant cells. Tight junctions join adjacent animal cells. Desmosomes join two animal cells together. Gap junction acts as channels between animal cells. So you see there are three kind of channels which are present in the animal cells, but in case of plant, only one channel is present that is called plasmodesmata. Now we have the plasma membrane. Plasma membrane is the outer covering of a cell. It covers the whole cell. It gives the cell a structure integrity. Just just like we all have skin around us, which is holding our blood, which is holding our skeleton, which is holding our muscles inside a body. We need a plasma membrane around the cell to hold its organelles and hold its fluid-like material. So a plasma membrane is composed of lipids, proteins, and glycoproteins. As I have shown in some previous slide like this, I said this is the plasma membrane, and this structure, these are the lipids, and this light blue color, this is the protein. So a cell membrane is composed of lipids, proteins, and glycoproteins. A cell membrane, there is a model which explains the structure of cell membrane. This model is called fluid mosaic model. So fluid mosaic model is what it says that a cell membrane is not a rigid structure as in case of cell wall. A cell membrane is a fluid-like structure. It is a fluidity. It appears that this is some fluid structure. This is not a hard structure. And it is in the form of a mosaic. Mosaic means different kind of things attached together, like in this image, there is a protein and there are lipids. So proteins are like mosaic. Proteins are like embedded in a proteins are randomly embedded within the cell membrane in an ocean of lipids. So there are like this everything is a lipid here, lipid here. This is the protein. This is the protein. This is the protein. This is the protein. So like proteins are in the form of a mosaic inside the cell membrane. And these structures which you can see here, what are these? These are carbohydrates. These are carbohydrates, and they can be cholesterol. They can certain other molecules which are present inside the cell membrane. So we have lipids as a major component of cell membrane. We have proteins embedded randomly within the mosaic in the cell membrane in the form of mosaic. So that's why it is called a fluid mosaic model. The plasma membrane is a phospholipid bilayer with embedded proteins. There are other components such as cholesterol and carbohydrates which can be found in the membrane in addition to phospholipids and proteins. These are phospholipids, these are proteins, these are carbohydrates, and these yellow crystal-like structures, these are cholesterol. So a plasma membrane has lipids, proteins, and carbohydrates, and it functions to allow selective passage of certain materials in and out of the cell. Plasma membrane functions. It is the boundary of the cell with many functions. It causes selective permeability. So plasma membrane has that authority that it can select whatever thing can come inside the cell or whatever thing can go outside of the cell. It is the plasma membrane which has to decide what is to be taken inside the cell and what is to be transported out of the cell. So how does that happen? There are there is charge on every molecule. Some molecules are positively charged and some molecules are negatively charged. So a plasma membrane has negative charge on it. It has a phosphate backbone which gives it a negative charge. So negative repulses negative. So whenever some this is the inside of the cell and this is the membrane. So whenever certain molecule wants to cross the plasma membrane, it needs to have a positive charge. It doesn't need to have a negative charge because cell membrane is already negative. It will repel anything that is negative. So substances which have positive charge on them will easily pass through the cell membrane. Moreover, this is lipid. What are lipids? Lipids are fat, and lipids work on the principle of like dissolve like. If there is a lipid molecule, it will only get dissolved in another lipid molecule. Just like you mix water with cooking oil, it will not get mixed. So this is the same principle here. So molecules which are like lipid soluble, they can easily pass across this membrane, but molecules which are not lipid soluble, they have the difficulty to pass through the membrane. So that's what we call plasma membrane has selective permeability. It allows some substances in and some are out of the cell. Moreover, plasma membrane provides immunity. Why? Because plasma membrane it distinguishes between self and non-self things. So plasma membrane has a function where it can select between the incoming proteins or incoming molecules, whether it belongs to the cell or whether it is some bacteria or virus. So there are certain complex mechanisms on which this immunity is provided by cell membrane. There are certain molecules on the surface of the cell membrane which help recognizing the antigens and proteins that self as self and non. So it is significant. Why is the immunity and self and non-self recognition by the cell membrane is significant? Because it helps in blood transfusion. Whenever you see, whenever some person has blood group A, it will only get transfusion from another person who is having a blood group A. Obviously, because there are cells in blood whose cell membrane function to recognize uh, self and non-self. Like if a person is having A blood group, so whenever he is given a blood group that is B, the person with blood group A, his cell membrane will recognize the blood group B as non-self, and it will not receive that blood. And then there is organ transplantation. You see, whenever there is an organ transplantation, like liver transplant, there is kidney transplant, certain other organ transplants, they are taken from the person who is compatible. Why is that? Why is that necessary? Because we do not receive things that are not like our body, and that is the function of cell membrane. Then comes a viral bacterial infection. Cell membrane also provides immunity against the viral and bacterial infection because cell membrane can recognize bacteria. Whenever bacteria knock on the door of a cell, the cell membrane will be asked who's there. The cell they will go bacteria. So the cell membrane will say, no, I do not take bacteria inside me. Just go away. So that's how cell membrane give us immunity. Cell membrane is, as we have already studied, it is a fluid mosaic model. So in this way, it can change its shape. So human cells, they need flexibility. They need a certain amount of movement from one place to another. They need to be adjusted. So that's why they need to change their shape every now and then, which is a function of plasma membrane. Plasma membrane, as it is a fluid structure, it is not a solid structure. It helps in the changing of shape of a cell. Immune cells squeeze between the blood vessel cells and go to the pathogen location. So, as I already discussed earlier, that whenever there is an infection on certain part of the body, the whole of the blood cells will keep on moving toward that part because we need some immunity there. So, immunity providing cells will be transported in the blood vessels, and while passing through the blood vessels, these cells need to change their shape to pass through the narrow blood vessels, and that is the function of plasma membrane. Then an important topic that is surface receptors. What are surface receptors? These are different kind of molecules, different kind of receptors over the surface of on the surface of cell membrane. And what is the function of these surface receptors, which we have already discussed, like plasma membrane functions. What was that? Selective permeability and distinguishing between self and non-self. How does plasma membrane achieve these functions? This is because of these surface receptors. So, for example, in case of HIV, when HIV virus comes, it recognizes certain receptors on the cell membrane, and then it causes infectivity. So, HIV docks that and binds to the CD4 receptor. So, this is the name of a receptor which presents on the cell membrane of T lymphocytes which receive HIV causing virus. CD4 receptor is a glycoprotein on the surface of T cells before entering or infecting the cell. So you see the significance of these receptors. They can even cause certain diseases as well, because some bacteria do recognize the receptors and they change the receptor. You can see the receptor shape and they can attach themselves with those receptors, and then the disease will happen. So, apart from providing immunity and apart from providing self and non-self kind of privilege to the cells, cell membrane also helps in the transport of certain materials. As we have already discussed, that uh, rough endoplasmic reticulum has ribosomes. Ribosomes, they produce proteins, and proteins are then transported to rough endoplasmic reticulum. And from rough endoplasmic reticulum, proteins are then passed to Golgi apparatus, and then from Golgi apparatus, proteins are passed outside of the cell via cell membrane. So cell membrane helps in the transport. So there are different kinds of transport, like passive transport and active transport. A passive transport does not use energy. Passive transport, molecules are moving from high concentration to low concentration. So this is a general rule of movement. Like whenever there is movement of a fluid, whenever there is movement of some salt, some solute, everything moves from a place of high concentration to low concentration. This is passive movement. This is passive transport. Then comes the active transport. When our active transport is what? It uses energy. Why does it use energy? Because it has to transport certain molecules from low concentration to high concentration. You can understand this concept from this example. Whenever we have water coming from a higher up place where water is more in quantity, so water will flow from higher concentration to lower concentration. But what if we need to move water from a low concentration area to a higher concentration area? We will be using certain motor pumps. So we need energy to transport things from low concentration to high concentration. And active transport is done via pump. Passive transport is it can be diffusion, it can be osmosis, and it can be facilitated diffusion. What is diffusion? A diffusion is the transport of substances or solute when in solution. Diffusion means transport of substances from an area of higher concentration to an area of lower concentration. And what does osmosis mean? Osmosis is same as diffusion, it just signifies the movement of water molecule. It particularly deals with the movement of water molecules. Osmosis is transport of water molecules from an area of higher concentration to an area of lower concentration. And then comes the facilitated diffusion. In facilitated diffusion, we use certain kind of membrane proteins. What does these membrane proteins does? It helps in the transport of solute from moving from one place to another. And these facilitating proteins can be channels or they can be transporter molecules. So there are three types of passive transport. One is diffusion, the other is osmosis. Osmosis is the same as diffusion, except it particularly deals with the movement of water. And then there is facilitated diffusion in which channels and transporters help. A solution is composed of solvent plus solutes. It has a solvent just like water and it has solutes. For example, if you mix salt in water, salt will be the solute and water will be the solvent. This diagram shows you the diffusion across a cell membrane. So you see there, first we had a lot of molecules. It means this was the high concentration area, and there was no molecule. So this was the area of low concentration. So with the passage of time, these molecules start moving toward the area of low concentration. See, now there are three molecules. Previously there was no molecule. So these keep on transporting from the area of higher concentration to the area of lower concentration. Eventually, the concentration of this side of the cell and this side of the cell became the same. This is called diffusion. Movement of molecules from an area of higher concentration to an area of lower concentration until the concentration on both the sides is equal. This is called diffusion. Then come the osmosis. Osmosis is the diffusion of water molecules until the concentration of the solute is equal on both sides of the membrane. It is the direction of water flow. But water flows from high concentration of free water molecules to low concentration. or from low concentration of solute molecule to high. Okay, you have to understand this concept. Let's suppose we have 2 glass of water, okay, and we dissolve uh, 5 ml of 5 milligram of salt in 1 glass of water and 10 milligram of salt in the other glass of water. So, in which glass there is high concentration of solute? Obviously, where we have dissolved 10 milligram of salt because it is more, so the solute will be more here. So, water will move from an area of high concentration to low concentration for water molecule. Whenever we are dealing with simple water, like in plants, we see plants in soil we have more water, and in the leaves we have less water. And water moves from soil to the root up to the leaves. That's because this is osmosis. Water moves from an area of higher concentration of water molecules to the low concentration water molecules. But whenever we dissolve solute in the water, now the concentration of solute matters. So water will move from an area of low concentration of solute towards the area of higher concentration of solute. I hope you are getting my point. It's the same as water going from an area of high concentration of water to the area of low concentration of water. But when it comes to solute, the water will move towards the place where there is high concentration of solutes and low concentration of water. This is osmosis. May I answer your question,
Thank you,
Dr. Visadia?
Dr. Mohammed. Thank you very much. Just entropy. This work that the cells are doing to keep themselves alive, to keep a certain amount of order, whether they're keeping something out or keeping more of a substance in, it's kind of like a constant battle to fight entropy. The total disorder. Everything tends to go towards uh, gradually towards entropy, and I guess is it the cell's job to keep you know uh, the opposite of entropy, basically, to keep itself alive, I guess.
Yes, and this is a proper process and a proper topic that is called homeostasis. You are using the word entropy, and for the cells, for the humans, we use the word homeostasis. Homeostasis means a specific and constant concentration of solute and solvent inside the cell. So the body tries to keep its cells in a constant amount of solute and in a constant amount of solvent. That's why it needs to transport certain things out and certain things inside to maintain homeostasis. So this is the process of homeostasis what you're talking about. And this is also called osmoregulation, particularly if we are dealing with the movement and transport of water and solutes. This will be called as osmoregulation. So the cells need to regulate the amount of water and the amount of solute in them. This is called osmoregulation.
Thank you.
And if we talk about temperature, this will be called as thermoregulation. So these are different kind of homeostatic mechanisms like osmoregulation and thermoregulation. And if possible, we will be able to discuss this homeostatic thing in the next lecture for sure. Now coming to the details of osmosis, solutions with higher concentration of solute are called hypertonic. Solutions with lower concentration of solute are called hypotonic. And solutions with the equal concentration of solute are called isotonic. And what do you understand from this? If there is higher concentration of solute in a mix in a solution, that will be hypertonic, and if there will be less solute, there will be hypotonic. So it's always the amount of solute which gets to decide whether the solution is hypertonic or the solute is hypotonic. And if there is, for example, I have two glass of water, and I put 10 milligram of salt in one glass and 5 milligram of salt in the other glass. So, in which the 10 milligram is added, there will be a hypertonic solution. And in the glass where I have put less amount of solute, that will be a hypotonic solution. So, what is the reaction of H2O flow in terms of tonicity of solution? As we have already discussed, that water moves from the area of high concentration to area of low concentration of water molecules. Uh, okay, Tafsile, we have a question here.
Hi, Thabsile. Please go ahead and join us. Just unmute yourself. There we
Um,
go. Perfect.
hello, hello, Dr. Mohammed. Hello.
Yes, we can
Hello,
hear you
yeah, I
perfectly.
can hear
Can
you.
you hear me? Okay, thanks. Some of us we struggling with blemishes, acne, pimples, etc. So, what might be the problem with our cell components? Is it the lysosomes? Is it the cell membranes which are not working properly? What is the cause of that?
Okay, there are different causes of acne and blemishes, but on the cellular level, if we can discuss, this is the defect of structural integrity, which is destroyed by bacteria, acne vulgaris. So acne is specifically caused by a bacteria, and this bacteria resides on the skin of specific people, and this bacteria destroys the structure of outer skin cells, and then they invade deeper inside the cells. That's why we do have blemishes. And this is basically cells die wherever there is acne, wherever there is acne scar, wherever there is acne mark, the cells are dead basically. So the dead cells appear darker and dyspigmented than the rest of your skin. That's why we get dyspigmentation, and that's why we get blemishes, because the blemishes you see are actually the dead skin cells of your body.
Excellent.
Okay, so we'll be going on with this image. So this is a hyper. Okay, so previously I was discussing that what happen if we put a water in which direction the water will flow. The water will flow from an area of low concentration of solute towards an area of higher concentration of solute. Like it will go from the glass where we have put 5 milligram of salt towards the glass where we have put 10 milligram of salt. So talking about the direction of flow of water, if we put some cells in a solution, for example, this black is a solution and this is hypertonic, and we have placed some human cells inside this hypertonic solution. So where does the water will move? The water will move out of the cells in this solution. Why? Because water moves from low concentration of solute towards the high concentration of solute. As this black solution is containing higher concentration of solute, the water will move out of the cells and the cell will get shrunken. As you can see here, the cell structural integrity is lost because the water has moved out of the cell due to the hypertonic solution surrounding the cell. And then there comes the hypotonic. If the black solution here, it is hypotonic, the solutes are like less in the water, but the solutes are like more in the cell. So what will happen? The water will keep on moving from this solution inside the cell, because remember the principle, water will always move from an area of a low concentration of solute towards the area of higher concentration of solute. So if wherever there is higher concentration of solute, the water will move towards that place. In this case, the solute concentration was higher inside the hypertonic solution. So water moved out of the cell, leaving the cell shrunken. In this case, the solute concentration is less in the outside environment. So that means there is more solute inside the cell. So water kept on moving inside the cell until the cell got burst. As you can see here, whenever there is a hypotonic solution and we keep a cell inside a hypotonic solution, the cell will keep on swelling and swelling and swelling until it bursts out. And then there is osmosis and plant cells. The same principle. If we place it in hypertonic solution, the water will move out. If we place it in hypertonic solution, the water will move inside the cell and it will cause swelling and bursting of the cell. But the plant cells do have cell wall, so bursting is prevented, because we have already discussed that plant cells have cell wall, and cell wall is a rigid structure. It provides structural integrity to the plant cell. While in case of animal cells, the cells got like bursted out because it didn't have any cell wall around it. So if we place animal cell in a hypotonic solution, the cell will keep on absorbing water and it will burst. But in case of plant cells, first of all, water will enter the cell and it will get accumulated in the vacuole. In the cell, we do have a vacuole where water can be collected before it starts accumulating inside the cell, like cell cytoplasm. But it will not get burst because cell has that plant cell has a cell wall. Okay, Dr. Ricardo, what's your question?
For hypotonic situations in the human cells, does that happen when the kidneys stop filtering water correctly out of the body, or what situations would that happen for humans?
Mm, Yeah, that's basically the function of the kidney to keep the solute concentration and the water solvent concentration in our body regulated. That is also a part of homeostatic mechanism. I already told you. This is also a part of homeostasis. So our body cells, yeah, our body cell needs to maintain an isotonic concentration of solute within its extracellular material. Otherwise, our cell will get either burst or our cell will get either shrunken. So yeah, that's necessary for the kidneys to keep the solute concentration right. Okay, then comes the active transport. Active transport is moving molecules against their concentration. So as compared to passive transport, we are moving certain molecules from their area of low concentration to the area of high concentration. So this is called active transport. But how does a molecule can move from an area of low concentration to an area of high concentration? It is because they use ATP as energy. ATP is provided by mitochondria, and we get energy from food. So that energy is used for active transport movement of substances from the cell membrane. Proteins in the membrane called pumps can use ATP to push chemicals from low concentration to high concentration. For example, sodium potassium pump. So remember when we studied the fluid mosaic model of cell membrane, in which we had like an ocean of phospholipids, and we had certain proteins embedded in the cell membrane. So those proteins act as pumps sometimes. They pump certain molecules from the area of low concentration to the high concentration. One example is sodium potassium pump, which we will be discussing in detail later on. So how does this active transport happen? There is an electrochemical gradient. And what does the electrochemical gradient mean? It means the total number of amount of solute and the total number of charges in a place needs to be constant. For example, if there are more positive charges on this side and there are less positive charges on this side, so for the active transport to happen, less charges from here will move toward the more charges here by the process of active transport. So that's why electrochemical gradient is important. It helps in the active transport of molecules. So electrochemical gradient arise from the combined effects of concentration gradient and electrical gradient. So what does concentration mean? Concentration means the exact amount, the exact mass per unit volume of a substance in a place. And what does electrical gradient mean? It means the ion, the charge on the certain molecule. Here we can see active transport. There are some, this is plasma membrane, and this is the phospholipid layer. And these are the embedded proteins. Here, another protein and another protein. And look at the direction of opening channel of this protein, like this. This facing this molecule is facing towards here, and this molecule is facing toward here, this molecule toward there, and this molecule here. And how did this active transport process go on? There are these molecules. They want to pass out of the cell. They came here, but this protein channel did not allow them to pass. Then they came out of this and they came toward this cell and they asked him, "Hey, can you let us pass through the cell?" And they say, "Okay, I will let you pass through the cell, but that will be using ATP." So then ATP came to the game and it provided energy to the molecules, and then this protein molecule help these little whatever molecules they are out of the cell. So that's how active transport works. It works with the help of ATP. And from where does the ATP come? It comes from mitochondria, because mitochondria are powerhouses of the cell. And what is the bulk transport? As the name indicates, bulk transport means the movement of big particles, like movement of fragments of cells, movement of fragments of organelles, movements of the whole organelles, like dead lysosomes, like macrophages, which are fighting against bacteria. So the movement of big particles is called bulk transport. And there are two processes involving bulk transport. Either one is endocytosis or the other is exocytosis. So see, if there is like this is the protein channel and this is a protein channel, and certain sized molecules need to be passed from here. As we already discussed that this plasma membrane is selectively permeable. It will allow those molecules to pass through it which have certain size and certain charge on them and certain solubility. It is highly selective. But for what say, let's say there is a molecule or there is a organelle which is this much bigger. How is that gonna pass through this cell membrane? This is a tiny cell membrane. So how is the thing which is this much bigger in size will pass? This will pass through bulk transport, either through endocytosis or exocytosis. Endocytosis means the movement of any molecule from outer environment inside the cell, bringing them into the cell. And what does exocytosis mean? It means taking them out of the cell. So exo means taking them out of the cell, endo means bringing them into the cell. So endocytosis means like phagocytosis and pinocytosis, and there is other receptor mediated endocytosis. So phagocytosis means bringing inside the cell those molecules which are solid. This is called phagocytosis. And pinocytosis means bringing inside the cell those molecules which are liquid. So phagocytosis is when big molecules or particles are brought inside, and pinocytosis means when large amount of liquid is brought in. And receptor-mediated endocytosis is when only specific molecules enter the cell. So this is all about bulk transport. And this is a diagram showing endocytosis. See, this was the cell membrane. This was the cell membrane. A large particle came. This cell membrane invaginated like this, and it like made a cover around this large particle. Then this membrane will fuse from here to here, and the cell and a vacuole will be formed, and the thing will be transported inside the cell, and the cell membrane will repair itself again. So a cell membrane pinches inside, it makes a covering around the big particle, and that big particle along with the cell membrane covering is transported inside the cell and as in the form of vacuole. This is called endocytosis. In the same way, when a thing is moving out, the cell membrane will bulge outside like this. It will bulge like this, and it will form a covering around that particular large particle, and it will pinch off, and then the cell membrane will repair itself. So this is how endocytosis and exocytosis work. This is pinocytosis, and this is receptor-mediated endocytosis shown here with the help of receptors. Three variations of endocytosis are shown. In one form of endocytosis, phagocytosis, the cell membrane surrounds the particle and pinches off to form an intracellular vacuole, just like this. In another type of endocytosis, pinocytosis, the cell membrane surrounds a small volume of fluid and pinches off, forming a vesicle, just like this. The only difference is this was solid and this was liquid molecule. In receptor-mediated endocytosis, uptake of substances by the cell is targeted to a single type of substance that binds at the receptor on the external cell membrane. So yeah, we studied about the receptors previously. Cell membranes having different kind of receptors. So a particular substance which gets attached to this receptor will be transported inside the plasma inside the cell. So this is an electron scanning electron microscope radiogram of a cell, which is showing endocytosis. Or this is how this was the cell membrane. It started bulging out like this, and then it bulged out like this properly. A proper bulge you can see in the cell membrane, and then it pinched off from the cell like this. A part of the cell membrane was separated from the original cell membrane, and it is containing now the molecules which are need to be transported. And then it went away, and the remaining cell membrane will repair itself. So this is exocytosis. I already described it in detail, like the same as endocytosis. When something is needs to go out of the cell, it goes via exocytosis. The same way the cell membrane. This is the cell, and this is the cytoplasm, and this is the cell, an extracellular fluid. Okay? And the cytoplasm, there are like a vacuole will be formed around the proteins to be transported. These vacuole will then fuse with the cell membrane, and the cell membrane will then open up outside environment, and the molecules will be released in the extracellular. In exocytosis, a vesicle migrates to the plasma membrane, binds and releases its contents to the outside of the cell. This is called exocytosis. And here we come at the end of our today's presentation. Thank you so much, guys. If you have any questions, you can ask them one by one.
Well, there you have it. Fantastic presentation by Dr. Muhammad. Um, Dr. Muhammad, I think we've learned a lot today from you. You gave us extra time, I think. Thank you for that. We covered all aspects of the cell, and what I'm most amazed at is all the things that can be learned and applied to so many other things. Here in the chat, we even mentioned, here at AIU, we focus on the UNESCO 2030 goals, how we can contribute to them. As you were talking, Dr. Mohammed, about the um, one of the topics of the cell, I was reminded of a recent article that I shared here in the chat, where we're actually creating plants where the leaves have a luciferase enzyme that's found in these insects. Scientists have been able to put it inside the leaves of the plant, and with that, they're able to create for 4 or 5 hours at night light up plants that are bright enough that you can read. So imagine being able to power the lights of cities at night without electricity. so, so many things we
That's
could,
so cool. That's so
yeah,
cool. Interesting.
there's so many things we can learn from this topic, from cells, because they've been evolving for millions and millions, billions of years. So
Yeah, yeah.
a lot of answers to everything are there: engineering, products, all kinds of things. So thank you so much.
Mhm. Thank you so
We
much. Thank you so much.
thank you for joining us, and we have one question from Tabsile. Please go ahead with your question. You're the only one so far.
All right, thank you, Doctor. Mine is about osmosis, the hypertonic situation. Like if you're suffering from, if you are sugar diabetic, do your cells sometimes do go through these situation hypertonic if you are sugar diabetic? And then if they do, how to correct that? If now they've lost all the water, they've become smaller, then how can you correct that?
Okay, that is simple. If you have a hypertonic situation inside your body, that can happen sometimes when your kidneys are not functioning or when you have diabetes. You have that increased amount of solutes and less amount of water. This is actually called dehydration. So you can simply rehydrate them. You can give them enough and plenty amount of water to rehydrate them. So we will be having increased amount of water, and automatically the solute concentration will get low. And on the other side, if we are having a hypotonic state in which the fluids are very low and body fluids are really high and water is really high, that condition is called edema of body. So edema is treated by giving drugs that are diuretic drugs. Those diuretics does what? They increase the urine output of the body. So you will be releasing more and more amount of water from your body, and your hypotonicity will be corrected. So that's how you do it. In hypertonic situation, you rehydrate the patient, and in hypertonic situation, you give them diuretic so they can urinate as much as they can.
Fantastic. Um, well, I'd like to do one more call for any questions today from our participants, our students, our special guests. Um, Otherwise, I'd like to once again thank you so much and to officially invite you, Dr. Mohammed. Hopefully you can make time in your busy schedule to join us again sometime soon and continue down this path of learning and discovery.
Yeah, I'm pretty sure I'll be joining soon, pretty soon. I will be joining this soon, and we'll be discussing some more topics, as you say. And yeah, we will be doing that.
Fantastic. Well, thank you so much. And thank you so much, everybody. So happy to see you. Happy holidays, whether it's Ramadan, Passover, Easter. Celebrate, have fun. Thank
You are
you for
such a
joining
nice
us.
Thank you
person,
very much,
doctor.
Thank you so
Dr.
much.
Ricardo.